Welcome back. So we are basically well deep into the topic of gene expression. And I think by now you have a basic understanding of some of the processes that occur in gene expression. The central dogma, just to do a very brief review, is we have something in the nucleus referred to as DNA. And we know, of course, that the DNA, through a process referred to as transcription, is going to code for a molecule known as RNA. And I think you have a basic understanding right now that there are several types of RNA that we have. We have something called messenger RNA, which is the transient carrier of the genetic information from the DNA that goes from the nucleus into the cytosol where the ribosomes are. We have transfer RNA, which is the direct carrier of amino acids to the ribosome. We have ribosomal RNA, which is the catalytic unit of the ribosome. We also have some other ones like small nuclear RNAs, which are basically RNAs that comprise the catalytic part of the spliceosome, which helps to do another process. I'll call this 1A, which is called RNA processing, and we basically for this we're processing the mRNA, although there is some processing for tRNA and so forth. But RNA processing occurs, and once we have all this business right here, we can essentially do another process, which is translation, which occurs at the ribosome, and we can get something referred to as a protein. Now, something that you've probably heard since you were in general biology is when you have a protein, it's comprised of 20 amino acids, and we can go into the long list of those, but suffice it to say, when you normally consider amino acids at the undergraduate level, you consider 20 of them. Well, it turns out that if you were looking at this absolutely, this is actually not correct. Okay, there's actually a few other amino acids that we can find in proteins, and actually what we're going to do is we're going to look at a specific one referred to as selenocysteine. Okay, and we'll actually look at a few of these structures up here. So let's talk about them a little bit. Well, right here, hopefully you can recognize some of these amino acids. Well, this one, all of these amino acids, of course, have the same backbone. But if I look at the R group of this one, it should be pretty obvious to you at this point that this one is L-serine. So I've, of course, drawn these all as the L isomer. And what I can do now is I can look at this amino acid. I notice it's a CH2 and then an SH. So this one has to be L. This is L-cysteine. But now here's one that you probably haven't seen. Maybe you have seen it and we don't really have an understanding of it yet. This one, instead of having an oxygen or a sulfur, the atom right there is replaced with selenium, so this one is selenocysteine. Okay? And this particular amino acid is often found in a lot of proteins that do things like redox reactions. So, for example, enzymes that deiodinate molecules like thyroxine, these have selenocysteine in them. There are some other ones we'll talk about in the next video that have selenocysteine. So suffice it to say, selenocysteine is very powerful in terms of redox reactions, and it's able to do ones that require a high redox potential. Okay? But what you should notice about this atom right here, and here it's the oxygen, here it's the sulfur, here it's the selenium. If you were to look at the periodic table, what you would find is that, for instance, I have carbon here, here's boron, here's nitrogen, oxygen, fluorine, and we usually neglect the noble gases. But if I numbered these, this is group 3, 4, 5, 6, and 7. If I go down group 6, I get to sulfur, and then I get to selenium. And these three are generally the only ones we're concerned with in that group in the case of biochemistry. And so you go down this group and you find they're all in the same group. And what that essentially means is they're going to have similar properties because they're in the same group on the periodic table. Okay? But it turns out that the selenium is even more of a powerful redox amino acid than sulfur, and oxygen really isn't at all. Okay, but as you go further in this direction, they become more powerful redox agents. Okay, now you look at selenium, and you might think, okay, Let me offer maybe a theory on how it might form. Let's say I have a serine. Let's say I have serine. Let me draw it like this. And maybe theoretically, let's say I have a selenide ion, an anion, right? Of course, this has a few lone pairs on it. And I do the mechanistic steps in green usually. Maybe what happens, perhaps maybe this selenium does a nucleophilic attack on this carbon, and then maybe you get loss of a leaving group, and the leaving group is essentially water. And if you did that, then notice you'd end up with two molecules. Number one, you'd end up with selenocysteine, right? And then, of course, you'd end up with water. And that seems like an efficient synthesis, but it turns out that this is not what happens. This is not how you synthesize selenocysteine. It's actually a little bit more of a complicated process. Okay, so actually, if you want to make selenocysteine, it takes more than one enzymatic step. Okay, and the step that we're going to look at in this video is the step in which you synthesize this molecule right here, which is called selenophosphate. And certainly these two molecules, this of course would be the conjugate acid, this would be the conjugate base. My pen's kind of messing up a little bit, but I think you get the idea. This is the conjugate base. And notice something. Let me draw a normal phosphate. So if I draw a normal phosphate, notice that it does have what we refer to as tetrahedral geometry. So this oxygen right here is coming out at us. And then you would have normally the oxygen up here. Well, if you look at the difference between these two molecules, notice the only difference is this atom right here. This atom is an oxygen right here on the phosphate, and this one is a selenium. And that really is the only difference. But this selenophosphate, this is what we refer to as activated, my pen's messing up again, activated selenium. So it turns out if you want to transfer selenium onto anything, generally what you have to do is activate it. And so we're going to look at the synthesis of selenophosphate, and in the next video we'll see what happens to selenophosphate. So we're just looking at the synthesis right here. And the enzyme that's going to be making selenophosphate is an enzyme that's referred to as selenophosphate synthetase. So this is the enzyme that makes selenophosphate, and the term synthetase in the name sort of gives us a clue as to what one of the reagents might be. Well, recall that synthetases in general, they are ligases. They're ligases that use adenosine triphosphate. Sometimes they can use GTP, but in general they tend to use adenosine triphosphate, and this enzyme is no exception. And really what's going to happen, I think you'll see, is there's two main steps that are going to occur in this enzymatic mechanism. Okay, one of the steps that's kind of going on is the actual synthesis of selenophosphate. And then what's going to happen is we're going to generate a molecule called adenosine diphosphate, and in just a minute we'll see what happens to that molecule. But what we're going to do right now is we're going to look at the mechanism by which we synthesize selenophosphate. And again, that's catalyzed by selenophosphate synthetase. All right, so this SPS right here, that's just representative of the enzyme as a whole. By the way, this X right here, you normally don't see this because normally when I do these mechanisms, I put whatever residue that is, and certainly X is not a residue for an amino acid that we find in proteins. I don't know of an amino acid that uses X as a one-letter symbol. All this X means is that that residue is not known. So the mechanism in general seems to be a little bit understood, and that's the one we're going to look at here, but that initial nucleophile that we're going to see is not known. So there's question as to whether it might be a cysteine residue. That originally was the theory. It might be a cysteine residue, but currently it's not well elucidated. So we're just going to put it as X, and we're going to say that it in fact is the nucleophile. And so what ends up happening is you allow this adenosine triphosphate into the active site, and selenophosphate synthetase catalyzes nucleophilic attack excuse me, on the gamma phosphate of ATP. And what that's going to do is what we're typically used to seeing. It's going to generate, number one, a pentavalent intermediate or trigonal bipyramidal intermediate. And I'm going to neglect this particular intermediate because I think we know by now what happens. You generate the intermediate, the phosphorus oxygen double bond reforms, and I'm going to kick off as a leaving group this molecule, which is adenosine diphosphate. And we're going to see, I guess we'll do it right now, what essentially happens to that molecule. Oops. Okay, so this right here is adenosine diphosphate. What's essentially going to happen is we're going to hydrolyze the adenosine diphosphate. And this may seem unusual to you because if you looked at most synthetases, they do not do this. But kind of the general theory as to why it does this is keep in mind these bonds right here between phosphorus and oxygen, these phosphodiester bonds, are high in energy. So the theory is that the reason it hydrolyzes this is to release free energy, has a negative delta G. So this process has a delta G that is less than 0. It's exergonic, and that release of free energy drives the other reaction to completion. So what's essentially going to happen is you're going to get a hydrolysis of this phosphate off. So this oxygen right here is going to extract this proton, catalyzing hydroxide, nucleophilic attack on this phosphorus, this beta phosphorus, and that's going to initiate the formation of this intermediate right here, which is called a pentavalent intermediate. This is called a pentavalent intermediate. The geometry of this phosphorus right here is trigonal bipyramidal, so you might also hear it referred to as a trigonal bipyramidal intermediate. But like all of these trigonal bipyramidal intermediates, it's going to collapse. So this intermediate is going to collapse back down to reform the phosphorus-oxygen double bond. And that's going to expel, let me scroll down, that's going to expel this molecule off, which is adenosine monophosphate. That will get recycled. And you'll end up with this molecule, which is known as phosphate. Now notice, this particular phosphate is diprotic. Here's one of the protons, here's the other. So this is, the equilibrium actually does not favor this form. So what will end up happening is you will essentially end up losing a proton to solution. Okay, you'll lose a proton to solution once this exits the active site, and you end up with the primary candidate for the kind of phosphate we would expect to find at physiological pH, and typically we refer to that molecule as orthophosphate. You can also hear this referred to as inorganic phosphate. Okay, so keep in mind that reaction generally is thought to occur because we're attempting to release Gibbs free energy to drive this other reaction to completion. Okay? Now what we're going to see is the actual synthesis of the selenophosphate. So let's do that. So this oxygen right here is going to abstract a proton from this hydrogen selenide. This is the form of selenium that's going to enter the active site. This is generally how you find it at physiological pH in the partially deprotonated form. That's essentially going to catalyze nucleophilic attack on this phosphorus right here. And then you're going to generate another pentavalent intermediate, which is shown right here. This is a pentavalent intermediate. And notice that this pentavalent intermediate is still attached to whatever amino acid residue this is. Now what's going to happen is you're going to get collapsing of the trigonal bipyramidal intermediate. This selenium lone pair is going to kick back down, and that's going to form a selenium phosphorus double bond, therefore expelling this amino acid residue as the leaving group. And so what you end up generating is this very strange structure right here. Now what's going to happen is you're going to do something referred to as a tautomerization. You probably very rarely have ever heard of a tautomerization. You may have talked about it in organic chemistry. All a tautomerization is essentially is it's a repositioning of a double bond with the subsequent movement of a proton. So what I can essentially do is, let's say for instance I have the molecule acetone. Right, here's acetone. What can essentially happen is basically, not particularly in this mechanism, it's a little bit different, but essentially this oxygen can abstract this proton, causing this bond to form a double bond here, with these pi electrons ending up on the oxygen. And so what you get in equilibrium is this molecule right here. this molecule right here, this is acetone. In fact, these are both acetone, but this one right here is considered the acetone tautomer. So what did I get? I got a net migration of a proton and a double bond. I can essentially do the same thing here. So what will happen in solution is the selenium will essentially attack this proton, causing a phosphorus-oxygen double bond to form, and these pi electrons end up on the selenium. And so what I get is a protonated selenophosphate. Oops, scroll over too much. I get a protonated selenophosphate as shown right here. This is the conjugate acid form. However, what will happen in solution is that selenium will end up giving up that proton to solution, and you'll end up with this selenium, which now has lone pairs that are tied up in resonance to the whole selenophosphate. And what we'll see in the next video is how that selenophosphate is used to synthesize the amino acid that I drew over here called selenocysteine. So this is selenocysteine. We'll see how that's made in the next video. And actually what's interesting is I showed you this hypothetical reaction down here, and I mentioned that this is actually not what happens. I'm kind of losing control of my pen. This is not what happens, but it turns out that it actually is made from serine. And it's not just any serine, it's a serine that's attached to a tRNA. So a tRNA, serial tRNA. Now the amino acid is serine, but the tRNA is a special kind that's specific for making selenocysteine. That's another point. SEC is the 3-letter code for selenocysteine. So if you ever see this, this is selenocysteine. So it's a tRNA that's specific for selenocysteine, but the tRNA synthetase specifically adds serine to this tRNA. And we'll look at that in the next video. See you soon.